Good morning. Thank you for joining us for today's webinar, Does Organelle Shape Matter? We're going to get started in just a couple of minutes at 10 a.m. Pacific time. Thank you for joining us. Do you want to go ahead and use the chat in Zoom or in YouTube to introduce yourself? You can go ahead and do that. We'd love to see where people are watching from. The recording of this webinar is going to be available on YouTube on the Allen Institute's YouTube channel after the fact. So you can find it at youtube.com/Allen Institute, and you can find all of our education resources at alleninstitute.com or .org. Goodness, alleninstitute.org/learn. We're going to get started. So at any point in today's webinar, if you have questions, please go ahead and use the Q&A function in Zoom that looks like 2 speech bubbles to ask those questions at any point. No need to save them till the end. And we have plenty of time to answer those questions at the end of this webinar. If you're joining us on YouTube, you can use the YouTube chat. If you're watching the recording of this webinar after the fact, you'll be able to find a link to our forum at forum.allencell.org in the video description, where you can ask those questions and our staff will answer those for you after the fact. So I'm going to go ahead and hand it over to Kim, who is going to get us started with this webinar today. Kim?
Great. Thank you, Kaitlyn. Well, welcome everybody that is here today to hear about our virtual science lab and open lessons for biology educators. My name is Kim Metzler, and I'm with the Allen Institute for Cell Science. I'm really excited today to spend this time with you and walk you through some of the open source tools that can be applied to lesson plans, specifically for teachers, educators, and so forth. So I first want to just give you a brief background if you haven't heard of the Allen Institute or if you're new to what we do. The Allen Institute, we're a biological sciences nonprofit that's located in Seattle, Washington. And we have a research focus in multiple areas in neuroscience and cell biology, immunology, and we also support cutting edge research across the world through the Paul and G. Allen Frontiers Group. Now, all 4 of our institutes really share a common goal, which is tackling complex, broad, and difficult problems in fundamental science. And we have 3 core principles that I'm going to come to in the next slide: big science, team science, and open science. And our work generates large amounts of data, knowledge, and analysis tools, all which we make available to the public. And I'm going to discuss a lot of those today. Now, our research approach is guided by these 3 core principles, as I mentioned before: team science, which means that we rely on this interdisciplinary teams that work towards a common goal. Our big science means that we take on large-scale projects and generate massive, useful data sets. And so far, we have collected nearly 25 to 30 petabytes of data, and that number keeps growing. And lastly, our approach is open science, which makes the Allen Institute really unique in that we share all of our resources to the public on our website, which I'll go through in shortly, which includes not only our data and our analysis tools, but also includes our products that, such as our cell lines, our Allen Cell Collection, that I'll discuss in a little bit. So more specifically, as part of the Allen Institute for Cell Science, our goal is to understand cellular behavior. So we're trying to understand what a cell did, what it's doing, so what's the current state of a cell, and where it's going. So what will it do? And we believe this is possible by looking at the cellular organization and cell function, which is tightly coupled. So these 2 being tightly coupled, and we're using this information to create a state space of human stem cell signatures. And we're trying to tackle this goal by taking a holistic approach using 3D, a little fast. We're using 3D live cell imaging of the major structures and organelles of the cell. And we're coupling this approach with, or we're trying to analyze this and use this approach we had to build the tools to create and tackle these scientific problems. Okay, so looking at cellular organization, we're using key structures and organelles within the cell because they can tell us a lot about cell's function. And so we've created the Allen cell collection. And what this is a collection of gene-edited human-induced pluripotent stem cells, or HIPSCs, that contain a fluorescent marker that's labeling key structures or organelles within the cell. And so if you're interested in learning more about our Allen cell collection, you can scan this QR code, which will take you to that resource directly on our website. It really describes all the different lines that we have available, because we have over 50 now. And what you can see here are 2 images. We take high resolution 3D live images of our cell lines. We take thousands of pictures for each of our cell lines, so we can get a comprehensive view of the cell and the organelles within that cell, to start to build these predictive models. And furthermore, we try to capture the variability and relationships of the organelles with each other, so that we can understand normal, healthy state of the cell. And so when the cell becomes perturbed by a drug or a disease, then you can see a change in those organelle relationships. And so if I show you here these two images, the first one is a video showing cell division in real time using one of our tagged cell lines called alpha tubulin. And the video on the right is showing the nuclear envelope that surrounds the DNA. It breaks down in these stem cell lines as a cell undergoes division. So you can see 2 different processes happening in a dividing cell by using our tagged lines. So this valuable information helps us understand our scientific problems. And we have created this science workflow in order to generate these high replicate and highly standardized imaging data. So we start, as I mentioned, with generating these gene-edited stem cell lines, and then we ramp up the scale of culturing these cells in a standardized process to prepare them for imaging. And so we use a confocal spinning disk microscope to take high-resolution live 3D images of our cells. And we further process these cells using a process called segmentation. So we can segment these images and get single cell information. So you get an image of a single cell, and maybe the single cell and the contents within that cell, whether it's the mitochondria, the Golgi, or other organelles. And this prepares us to begin. This can prepare the images for downstream analysis, modeling, and we manage and store all this data. And I'm going to show you and walk you through. We put all of our imaging data up on our website and make it publicly available. And so this is called our Allen Cell Explorer. So on our website, we have a toolkit of a suite of tools to be able to visualize, model cell behaviors, and all of which are open source and available to the research community. We call this our Allen Cell Explorer Toolkit. And in this toolkit, I just mentioned the Allen Cell Collection, which is the collection of our 50 gene-edited cell lines. We have a 3D cell viewer, which contains our image collection of all of our 3D images. We have the Allen Cell Structure Segmenter that does the segmentation to help process and get those cells ready for analysis. And then we have our visual guide to human cells, which I'll talk about in just a little bit. And we also have our Cell Feature Explorer. But I want to first start by introducing our website, the Allen Cell Explorer. So when you get to the homepage, you can see a menu at the top. And in this menu, if you click on, say, education, which is relevant for this talk, you can see a bunch of resources listed below. And I'm going to walk you through these resources. So if I click on all resources, what you'll get is this page, educational resources with some links that will take you directly to certain specific materials or videos or other online tools. And if I just scroll down this page, you can see that there's, whoops, here. Let's see. Okay. You can see that there's some lesson plans that you can take you directly to certain lesson plans that, Carlos will go over a lesson plan today and how he incorporates it. But also you can see there's some links here for the CourseSource virtual lab. You can see here's Carlos and other teachers that have helped us create certain lesson plans. And there's more information below where you can get, say, a quick start guide for teachers. This quick start guide for teachers is a downloadable PDF. It gives you a really high level overview of each of our resources that I'm going to walk through with you today. So, for example, the Visual Guide to Human Cells. This is an interactive resource, and what it does, if you can see on the far left on the panel, it will display several different major structures of the cell. There's 14 total on this page, and it tells you about like the different functions. And so if I click on this, what you can see if I hit Sense the Environment, underneath you'll see 4 different structures. So microtubules, it'll take you to another link that shows you how microtubules look in different stages of mitosis. So prophase, prometaphase, metaphase, and anaphase. And you can go back to interphase. And this tool really allows, oops, this just went, sorry. This tool, what did I say? This tool is really good for giving an overview of the anatomy of the cell and getting students familiar with what the function of these structures, the function of these structures play in the cell. And it can actually provide even a more accurate view of the cell, because these are 3D renderings from our actual microscope images of the cell. There are some also, we've done some machine learning and applied some machine learning rendering to capture an integrated model of this 3D visualization, but it gives you a more real look at what a cell looks like and the structures within. Now, if I go back to these different tools, and if I click on the integrated mitotic stem cell, you can see here, this brings you to this page. And what the integrated mitotic stem cell is a visual essay that introduces a data-driven model and visualization tool that captures human cell division by integrating the 3D microscopy imaging data from a collection of 75 single cell images. And it goes through and walks you through the different stages of mitosis, and goes through other information up here on this toolbar that's at top. And so I'm not going to walk you through this whole essay, but to show you that this is here and available. But really, there is these important, I think, and valuable tools that are accompanied with this essay. And this one here is the Z-Stack viewer that biology teachers can use to describe different cell and organelle relationships and across all the different major stages of the cell cycle. So here is the stage of the cell cycle, and here's the different structures that we've imaged. And if I then show you what you can do is if I clicked on, say, the prophase and the nuclear envelope, then what you'll see here is this window pops up, and you can actually scroll through the Z-stack of this image and look at the nuclear envelope, which is this white structure here, and see how it comes and goes through the entire stack. So it's a fun tool to kind of actually get you familiar with microscopy images, familiar with the different structures of the cell, and really this different stages of mitosis. So now we also have in this same, at the end of this same, the toolbar, we have a 3D viewer. And this can be used to help also describe the different cell and structure relationships across the major stages of the cell cycle. But here it's a different way to view it. So here you could look at some of these, so you get to know the actual gene name or the protein name of the structure that is tagged in the cell lines. And so you can click on the different structural name, and here is the actual structure that it labels. And so right now I'm showing every structure is labeled in this example, and you can move that cell around. Oops, it's automated. So you can move that structure around, and you can
get that. Could you try again?
You can toggle it, and you can kind of remove some of these structures if you'd like to get a more, narrow it down or to a view that you really want to focus on. And so I think it's a very valuable viewer in that case, and really can help students learn about the different structures and the relationship to each other during the different stages of the cell cycle. Okay, now going back to our menu, we can find other valuable resource that can allow you to view the 3D microscope images that we've generated. There are two options here. You can either do the 3D cell viewer, and this contains data that we collected prior to 2018, and we revamped our viewer and renamed it the Cell Feature Explorer, but we still have the older generated data under the 3D cell viewer, and our newer data is on what's called the Cell Feature Explorer. And this is a new tool that includes an interactive plotting map, so you can compare different cell features within the cell. So I'll kind of highlight that here. Here's the page, landing page of the Cell Feature Explorer. Here you can see the top 2 data sets that are available. If you scroll down, there are 2 additional data sets. Here is 2 different single cell imaging data sets. If I click on the one on the right and load that, what you can see is, if I click on that and you can open that up, what you would see is the actual plotting tool and so forth. And I'm going to kind of demonstrate that right at the very end, but we'll see. So, and then finally, I want to just mention the going back to the educational resources page. There are ready-to-use activities that include for using that 3D cell viewer that I mentioned. So you can try our introduction to microscopy and biology, for example, on the left. This is really good and valuable for, say, high school level students, or our virtual laboratory on cell division that uses the publicly available image data set that I just referred to on the 3D cell viewer. So these are available on our website on this educational resources page. If you scroll down, you can get to them. And finally, I want to since I have introduced our tools of educational resources, I'm going to turn it over to Carlos Goller in just a second, who's applied our tools in developing a lesson plan for his students. He's an associate teaching professor in the Department of Biological Sciences and teaches in the biotechnology program at North Carolina State University. And I want to end by thanking our cell science team, who has generated the resources and tools that I showed you today. And before and so now I'll hand it over to Carlos to walk you through the lesson plan. So thank you.
Perfect. Thank you so much for inviting me. And this has been, over the last couple of years, has been a really great collaboration that has taught me, a microbiologist, several new tricks and data sets, and has impacted lots of students over the years. So I am going to hopefully share my screen correctly. Is it working? Yes.
Yep.
Okay. So a couple of years ago, in 2018, Sabrina Robertson and I were able to get funding from the National Science Foundation to start a collaborative network of educators and researchers to learn about high throughput approaches in the classroom through exploring real data sets, huge complex data sets using cutting edge techniques. And we reached out to the Allen Institute, and it started this fantastic collaboration. And Kaitlyn has been really an instrumental leader in getting us connecting the data sets with the classroom activities. And today I'll talk about, does Organelle Shape Matter exploring patterns in cell shape and structure with high throughput imaging. And my task as an educator and faculty member at North Carolina State is to teach in the biotechnology program, an interdisciplinary program where we have students from a variety of programs, majors, and educational trajectories. Some are in master's programs. We have graduate students, including PhD students and vet students working in the same labs and classrooms and virtual spaces, learning new techniques in molecular biology. So the biotechnology program teaches a variety of different majors and programs, and that's a challenge because we want to teach them hands-on data analysis and high throughput cutting techniques that often we cannot do in our lab space. So, and I'm sharing the slides in this link if someone wants to follow along. It's go.ncsu.edu/Allen 2022. So my charge was to bring high throughput technologies into the biotech labs. And our tagline was, do not fear the robot, do not fear automation, do not fear high content microscopy or high throughput screening. We can learn this together. So we used high throughput approaches and told participants in courses, these are becoming increasingly common and we have to learn how to manage them and analyze them. And for this, we wanted to introduce high content, high throughput approaches to students that may have limited experience learning about the methods and how the data are generated. So our hook was to create a case study that was engaging. Our approach was to include students in exploratory assignments. And using the pedagogy of case studies, we are able to present a scenario that's realistic. And with the open data sets and tools that are web-based from the Allen Institute, we put all this together and we're able to create what I would describe as a really dynamic and modular lesson that we published this year in CourseSource. So the lesson has some goals and objectives we set at the beginning of the creation process. And these are, first of all, we wanted to introduce students to how organelle structure varies and how we can use high content or high throughput approaches and data sets to investigate this question. And we focused on the ER, the endoplasmic reticulum. And also we wanted to encourage students to apply open data as a reference to provide context for their own data sets and their own analyses. Always we want to help support students through scaffolded lessons so that they go from remembering, understanding, applying, analyzing in Bloom's taxonomy to evaluating and creating. And we were able to do that with our lesson objectives. We started with describing the morphology and the anatomical structure of the ER, describe the purpose and applications of high throughput microscopy. Then we did some comparing of cell structures using the visual analysis tools on the 3D cell viewer on allencell.org. And finally, we challenged participants to design their own experiments using similar data and building on their findings that they accomplished through this case study. So the lesson itself, as an educator, I think about it as having three stages: the setup to build the foundation for why we're doing this lesson, and really motivating students to get interested in this topic and understand why it fits into the course goals. Next, we implement it. And finally, we need some way of evaluating our learning outcomes, so assessment and potential modifications. The setup on logistics was really neat for this case study because the lesson can be implemented in a variety of courses, including molecular biology, genetics, and biotechnology courses, both at the undergraduate and graduate levels. And participants can work individually and in groups. And this may have happened serendipitously during our shift to online remote teaching, but we were able to share this case study with other educators and implement it here at NC State in both online and in-person settings, and individually or in teams, in groups. And finally, the activity can be done both synchronously as well as asynchronously. And students can submit their work as hard copies or digital responses. So it's really modular and flexible. So first, we want to determine ways to distribute the handout and the resources, the case study scenario to students, and that can be electronic or hard copy. I often introduce the learning objectives and context for why high throughput microscopy would be done, who is doing it, what has been found with it, and why is it a cutting edge and necessary approach. So then we introduce the case study. If necessary, we create groups. We provide guidelines for the timeline for when students will work individually, when students will work in groups, when they will have access to the different parts of the case study, and when they will reflect and submit their final work, which is often the completed case study, all the questions and some reflection prompts. And finally, we reviewed the submissions, and it's really important for case studies to debrief with the class, with being in person or online through a video or an announcement. What did we learn? Why is this important? Review some of the responses students had. So in our publication, we shared a detailed table using the course source format that goes through the pre-class preparation and what happens in the class, introducing the case, during the case study, during the reflection, and during the debrief for an in-person typical lesson. So when we implement this case study, it has several parts. And I really love this because we started with a scenario, and I may have written myself into the case study as Dr. G, a scientist with grad students in his lab. And he's a microbiologist, and they are exploring some phenotypes they observe as they treat some eukaryotic cells with a potential antimicrobial compound. And the first part of the scenario is, okay, we stain these eukaryotic cells. We're observing their ER, and we notice that the ER is really different from what we expected. So part two is, this image does not look like the textbook image for an endoplasmic reticulum. What is happening? So then we engage students in the case study, and we have them observe different cells through the Allen Cell Viewer, the visual guide. And what's really powerful is that we're able to do some quantitative analysis and do those on the web. And finally, we ask participants, does this make sense? What would you recommend for Dr. G? So we begin with what happened to these cells. What does the endoplasmic reticulum look like? And does its structure matter? And we often have the high school image of the endoplasmic reticulum or the textbook image of the ER. And we contrast that to what the visual guide to human cells provides and how it's able to depict the ER over different cell cycle stages. For part 2, we ask, this does not look like the textbook image. Why? And is this important? So participants use the Allen Cell Explorer visual guide to human cells to learn about the methods researchers used to visualize the ER. So now we engage them and have participants think about the methodology and the techniques. So for example, in part 2, they navigate to the Allen Cell Feature Explorer. They select Sec61 beta under protein tags from the menu. And we have them visualize cells with fluorescently tagged Sec61 beta, which is for the ER. And we tell them, do you notice any patterns? And this is a sample student response. And they say the Sec61 beta protein tag is used to visualize the ER in different cells. So now they are comparing different images of cells. And that's what's really powerful about this large data set. In different cells, the ER fluorescence surrounds the nucleus, but the pattern does vary. So now students are noticing that variation. And in part 3, participants dig deeper. They begin to quantify cellular and nuclear volumes using the Cell Feature Explorer. And over here on the right, you can see on the slide how the cellular volume versus nuclear volume can be graphed, and students can click on each dot to view an individual cell and the data associated with that individual cell. So they are working with and interacting live on a website with thousands of cells. So then we have students work either individually or in teams to calculate the cellular and the nuclear volume. And this is a sample of student responses. And often if they are doing this individually or in teams, I have a Google Sheet or a spreadsheet we can put up on a screen or share so that students can compare their measurements, and then we have an opportunity to do some graphing and statistics. And in part 4, we ask, does this make sense, Dr. G? So students visit this website that the Allen Cell Explorer has prepared with a nice visual that we saw previously with Kim's presentation on how edited cells were created, how automation was used to take care of these cells, to visualize thousands of cells, how high throughput microscopy was used to take 3D time series, and how this data can be analyzed to create an open data set, as well as model and do some label-free predictions. Lastly, we asked students 2 important questions. What was the most memorable concept or skill you learned from this case study? And what are you left wondering about? And the only unacceptable answer is nothing. And the sub-question is, how do you start the process of learning this answer? And over the years, we've been doing this for now 4 or 5 years, every semester or every other semester, with classes ranging from 10 to 16. And the variety of answers we get for these reflection prompts is really exciting. So there's also an opportunity to modify this case. And going beyond the ER, this is a rich cell data set. So we can take this and branch off this case study and these data sets and these approaches to investigate several other concepts and skills that get to data analysis, get to computational approaches, get to cell biology, developmental, and even some molecular techniques. So for example, students are often interested in the label-free prediction system. And in one class, we spent some time reviewing the Nature Methods paper associated with this approach. And we talked about the importance of the training data sets and the data sets that were used to create this system. Students often want to continue exploring the data sets and tools in more detail. So for example, we've had groups of students ask, are certain structures different in the certain ER structures different in this type of cell versus this other type of cell, or this growth or cell cycle stage versus another one. And this is a rich data set, and I've mentioned this a couple of times, but it can be leveraged to spark curiosity and emphasize real-world applications from molecular to the data science perspectives. So I'm really excited about what we can do next with this case study and future data sets, including drug perturbations, including the use of CRISPR to create the cell lines. So I would like to thank the Allen Institute and the HITS Case Study Network for their patience. Going back and forth, we've reviewed, revised this case over several years, and we were able to modify it and make it work for a variety of different course settings, including from face-to-face to a hybrid course to a completely asynchronous offering of graduate and undergraduate course in molecular biology and high throughput techniques. So with that, I would like to thank you all and ready for questions.
Thank you so much, Carlos and Kim. We have some questions, and audience members, we have time for questions from you. We have a couple of questions have come in already, so please keep those coming. So to start, these images are very cool. I use augmented reality in my classroom. So I was wondering if it would be possible to explore the possibility of using images and visualizing them in AR. And perhaps I can prompt you to maybe talk about virtual reality as well as augmented reality. Kim?
That's a great question. So we have actually, we do have the capabilities of viewing these cells in augmented reality. We have put together some work towards being able to visualize our cells in AR. We also have a game that I did not mention that's called Mitosis, and it's on, it's made available as a VR game. If you have a VR headset, I will send you guys the link to that on the chat momentarily. So you're welcome to take a look at that if you're interested.
So this question is for both of you. What software do students need to download in order to do this lesson?
Oh, I can tackle that one. That one's a really easy one on my end, because honestly, I've used Google Docs to share the case study or Word documents or handouts, and all they need is access to the internet and the website. And the website has been fantastic because it works on several browsers. To this day, I haven't had anyone mention browser issues. And we've even embedded parts of the lesson into our learning management system as pages, or using clicker software. We use Top Hat as embedded questions with links to the website. So all they need for the calculations is access to the website and maybe a spreadsheet.
Yeah, no software downloads required. You can download the images from the website and open them using something like ImageJ, but you don't have to do that. You can analyze everything in the browser. Question for Carlos, and remember, audience members, we have plenty of time for questions from you, so go ahead and put those in the chat. This next question is for Carlos. What classes do you think this lesson would fit well with? And in your class, what other units does this connect to in the curriculum?
So that's a fantastic question, and I'm continuously impressed by how people have used the visual guide to the cell and other resources available at the Allen Institute. We have used this case study as part of a junior and seniors and graduate student 8-week lab module course that's focused on high throughput methods. So very specific. Yet I've heard of educators using this case that we've shared for genetics courses, for molecular biology courses, and cell biology courses too.
Great. So next question is for you again, Carlos. What sorts of things did you find your students struggled with in this lesson?
I think, and I'm thinking about this one, I think it's often having too much choices. It may be ironic, but they had too many cells to select which ones they wanted to measure. or sometimes we get distracted. So in person, when we did them in small groups, I would walk over to a group, and they were now investigating other organelles. And while that's really useful, they drifted from the case study a little bit because they were playing with the visual guide or playing with the cell explorer. And that was one challenge. The other challenge was to explain a little better the connection between the tools measuring cell volume, for example, and why would this make a difference? Why is this intriguing? And there are some really great examples of organelles and their size affecting function or being a function of environmental stresses, and including the ER, and bringing that into the conversation midway through the case study or prior to the case study was a good way to help students with that.
This next one is for Kim. What other tools does the Allen Institute offer to educators? And if you can specifically speak to the ones for the Allen Institute for Cell Science.
Yeah, of course. Yeah. So I mentioned some today, but the one that I didn't mention that is one of our newly released tools is called Simularium. Simularium is a visualization platform that allows people to observe different models and be able to and we're working towards being able to manipulate the different parameters of these models so that you can see what happens to, say, a molecular model as something has changed, something maybe environmental or internal to the cell changes. You can kind of see how that is affected. And this tool's the Simularium. It's on our website. It's basically a simulation tool that inputs simulation models from different softwares. And so people that are more advanced that are creating simulation outputs, they can actually upload their models as well. So that it does make it available for a more advanced expert that has their models that they've created. But I think it's a valuable tool in looking at these models and understanding, more biology, more complex biology by using these models. And we have a bunch of examples on our website, and they're linked to papers. And so you can actually, get an idea about some scientific concept from the paper, which is an advanced, a skill to evaluate a paper, but also get to play with the model on our website. So that would be one I would recommend for sure.
Yeah. So the resources that Kim mentioned are using open data and other tools from the Allen Institute for Cell Science. We also have education resources that connect to the research being done in other institutes within the Allen Institute. So I run our education program that spans the entire Allen Institute, including cell biology, neuroscience, and immunology. Those resources can all be found at alleninstitute.org/learn. And we have lesson plans and activities that connect to our open data, that connect to the papers that we publish on our research, that get students into the data like this lesson. We have more and get them in there and get them playing with it, getting them to do real manipulation, real experiments, really getting their hands into that data all online. Next question for both of you. What if we wanted to use Allen Institute resources and tools for student research projects? What do we need to do to have our students be able to use data for student research projects?
I'll first start off that, we've created this website to make it publicly available to students. If you do use it for your own research project and your professor or teacher, asks for a reference, you can cite the allencell.org, or if it's related to or connected to a paper or manuscript, you could cite that manuscript as well. I'll let Carlos answer towards, using these tools for specific projects in the classroom.
It's out there, and the data can be downloaded. So we've had students get interested in the drug perturbations data set and download lots and lots of images and do some scanning and quantifying. There are lots of possibilities here. And from the method side, I'm really excited about the label-free prediction system and as a way of bridging cell biology and modeling. And Simularium is going to be really cool. I was just thinking about ways we could use it. And microbiologists, there's an E. coli MinD model that's really neat.
This question is for Kim. How often does the Allen Institute for Cell Science provide new data on the website? And tools as well, actually.
Yes, that's a great question. So we post new things as we develop new data. We definitely curate and make sure that our data is at a good standard to release publicly on our website. But we often update our website quarterly or yearly, depending on what type of data is present. So for example, if we have new cell lines, we'll release that data and post an update on our website. So we'll always post an update on our website, and you definitely can sign up to stay updated on any new resources that we add or new content that we add to our website. And I think we can post that link on the chat shortly.
Yes, that link is in the chat. And also for those of you watching this after the fact, I'll get it into the video description.
Okay, thank you.
Carlos, how did your students respond to this lesson? You asked them to reflect on their experience. What sorts of things did they say?
They were really curious about the methods, which was exactly what I wanted them to focus on. We got several questions about what other cells have been image this way? How can the tools be used for research? Maybe for their own research, for undergraduates and graduate students doing research. The other thing that really intrigued people was, okay, so how are these cells maintained, and all the work that goes into creating the cell lines and maintaining the cell lines. And that's where, in our case, automation was something that we kept on going back to in the course. It's a course goal, and students really honed in to how the high throughput and the immense amount of data that was generated by this approach was done because of some automation tricks and lots of work and optimizing, but there's a lot of preparing, making this, creating the cell lines, verifying them, that goes into it, that I'm glad people reflected and found that to be important. And it came up in reflections many times.
So you mentioned what class you taught this lesson in, or classes, plural. What classes would you recommend this learning experience for? What levels of student? And how long does this lesson take in those classes? How many sessions?
I actually think this would be really well suited for a cell biology class at the maybe 200 or 300 level. And it could be done in as little as one period, maybe in groups, or you could assign part of the case, be it the background and maybe reviewing some things on the Allen's webpage. We have components that of the case that can be done asynchronously or as pre-work. And what's really important, I think, is how much time you spend debriefing or discussing the connections. And I think that could be done in one full class period, or maybe 1.5 if you give. I've had what I'll call good luck, but good responses and good student interactions, letting them, after they do the case study, coming back a week later and saying, now that you've had a chance, what was beyond tinkering with the website, what stuck with you? And I think that makes it come to Martina's question. I would say at least 50 minutes to do justice to the group work and the discussions, but our lesson can be trimmed. You can take some components out. The scenario is really adaptable.
Yeah, and adaptable to the comment in the chat about being a virtual lab. Yes, this can be done entirely online. The wonderful thing about using open data in teaching is that you can give students experimental experiences that are fully online if they don't have access to a lab because they're remote learners, or your institution doesn't have lab facilities, or doesn't have lab facilities that can collect this type of data, or doesn't have access to human data because these are from human cells. So yes, it can be adapted to be fully online, hybrid, part homework, part in class. There's lots of adaptations you can do. Yeah. So we are coming up close to time. Carlos or Kim, do you have any final thoughts you'd like to share about using open data for teaching, how this lesson goes in your class, anything else, just to wrap it up?
I would like to just add that, if you guys ever have questions about using our resources or have any issues, we'll help. We'll definitely help. We don't want that to be the bottleneck of you using it. If having some sort of issue on our website, if something's not working for you, we can troubleshoot, or if you just have simple questions, we're happy to help as well. So don't be afraid to reach out. That's a part of our open source and data sharing is to be open with other people too. So thank you guys for coming.
Yeah, you can contact us via at our homepage at alleninstitute.org. Scroll down to the bottom and there's a contact form. You can email us at communications@alleninstitute.org, or you can contact us at the forum at forum.allencell.org.
Yeah.
Carlos?
And I'll say that as students have mentioned, why is the Allen Institute sharing all this, or how long did it take to develop this and then share it? And it brings up some really important conversations about open science, about how we can share methods that, and how data sets may be shared, and then you can still revisit those and find new things and find ways of creating another method based on it, or use a subset of the data to investigate another question. So it really sparks great discussions that are important and may not be concepts in your class, but are important as developing scholars and researchers, and our job training them.
Yeah. Well, we are almost at time, so we're going to wrap it up here. Thank you, everyone, for joining us today. You can view the recording of this webinar on YouTube, so you can review this content after the fact. And again, if you have any questions about this lesson, our data, our other open education resources for our other divisions of the Allen Institute, or our other education resources that are related to the Allen Institute for Cell Science, please don't hesitate to contact us. And we hope we'll see you at future virtual events and other programs for educators. Thank you.